Today we discuss the thermophiles and its molecular adaptations. The thermophiles are the microorganisms that grow at high temperature of 55 degrees centigrade or more. Minimum range is 45 degrees centigrade and optimum between 55 to 65 degrees centigrade and maximum is 80 degrees centigrade. Some microorganisms grow even at more high temperatures, the optimum between 80 degrees and about 113 degrees, and are called hyperthermophiles. The latter usually do not grow well below 55 degrees centigrade. Thermus aquaticus, Thermoplasma, Acetophyllum, Bacillus stereothermophilicus, etc. are the examples of thermophiles, whereas Pyrococcus absci and Pyrodictum oculictum exemplify hyperthermophiles. Most hot springs have temperatures at or near boiling. Steam vents may contain temperature to level of 150 to 500 degrees centigrade. Hydrothermophile vents present in a sea bottom have temperature of 350 degrees centigrade or greater. Hot springs are found throughout the world, but are especially concentrated in western United States, Central America, Central Africa, New Zealand, Indonesia, Italy, Japan, and Iceland. The world's largest single concentration of the hot spring is in Yellowstone National Park, Wyoming, USA. Thermophiles and hyperthermophiles in hot springs and hydrothermal vents. Many hot springs are at the boiling point, and in them a variety of hyperthermophiles are typically present. The growth of such microorganisms can be studied by immersing microscope slides into the slide and retrieving them after a few days. Microscopic viewing of the slides reveal colonies of prokaryotes that have developed from single prokaryotic cells that attach to and grew on the glass surface. The boiling water overflows the edges of the hot spring and flows away from the source. It gradually cools, setting up a thermal gradient. Along this gradient, various thermophiles grow with different species growing in different temperature ranges. Large number of sulfur oxidizing chemolithotrophic hyperthermophiles are preset in the around sulfide emitting hydrothermal vents. Sample collected near such vents have yield cultures of Thiobacillus, Thiomicrospira, and Bigotia. A certain vent sites of deep sea superheated hydrothermal fluid is emitted at temperature up to 350 degrees centigrade. This superheated but not boiling water at depth of about 2600 meters in sea water does not boil due to huge hydrostatic pressure until it reaches a temperature of 450 degrees centigrade. Could theoretically be a habitat for hyperthermophilic bacteria. The hydrothermal fluid emitted from black smokers. A deep sea hydrothermal vent emitted superheated 250 to 400 degrees centigrade water and minerals contain abundant metal sulfide, especially iron sulfide, and cools quickly as it mixes with cold sea water. The precipitated metal sulfide form a tower referred to as chimney. It is found that the wall of the smoker chimney teem with hyperthermophilic prokaryotes such as methanopyrus. The most thermophilic of all prokaryote species, Pyrolobus and Pyrodictum, also reside in smoker chimney walls. Molecular adaptation to thermophiles. Following are the factors that help thermophiles and hyperthermophiles thrive at high temperatures. Enzymes and other proteins of thermophiles often differ to some extent in their amino acid sequence from enzymes that catalyze the same reaction in mesophiles. So mesophilic organisms are somewhat different than thermophilic organisms. It appears that a critical amino acid substitution in only a few locations in enzymes of thermophiles allow it to fold in a way that makes them much more stable to fit, and as a result, they function optimally at high temperature. Thermophiles typically possess lipid rich in saturated fatty acid in their cytoplasmic membrane, thus allowing the membrane to remain stable and functional at high temperatures. Saturated fatty acids form a stronger hydrophobic environment than do unsaturated fatty acids. Such hydrophobic environment help for the membrane stability in thermophiles. Heat stability of protein in hyperthermophile is also improved as a result of an increased number of ionic bonds between the positive and negative charges of various amino acids. This makes densely packed, highly hydrophobic interior of the protein, which naturally resists unfolding of the protein in aqueous cytoplasm. Also, solute of diinositol phosphate, diglycerol phosphate, and monosyl glycerate that are produced in high amount in the cytoplasm of certain hypothermophile help stabilize their protein against thermal degradation. Since most of the hydrothermophiles are archaebacteria, they do not possess fatty acid at all in their membrane, but instead contain lipids having branched C40 hydrocarbon chains composed of repeated units of 5-carbon compound called isoprene bonded by ether linkage. The ether linkages provide more stability to membrane against thermal breakage, and the branching of the hydrocarbon chain decreased membrane fluidity. Therefore, the membrane of hyperthermophiles are stable under high temperature conditions. Additionally, the overall structure of the cytoplasmic membrane of hyperthermophiles form a lipid monolayer, and this structure is undoubtedly much more heat stable in comparison to lipid bilayer of species of bacteria and eukaryotes. Biotechnological applications of thermophiles. Thermophilic and hyperthermophilic microorganisms often offer some major advantages for industrial and biotechnological purposes, many of which run more rapidly and efficiently at high temperature. Exchemozymes and molecular chaperones obtained from hyperthermophiles are the examples. Exchemoenzymes. The term exchemoenzyme has been coined to describe enzymes that function at some environmental extremes such as high temperature and low pH. Since most of the industrial processes operate best at high temperature, exchemozymes from hyperthermophiles are widely used in both industry and research, and such exchemozymes are proteases. Proteases, amylases, glucose isomerase, cholineases, xylinases, glucoamylases, Taq and PFU DNA polymerase, glutamate synthetase, and aminotransferases. All these are the examples of exchemoenzymes. Proteases. Proteases, that are protein hydrolyzing enzymes, have been isolated, purified, and characterized from Pyrococcus, Thermococcus, Sulfolobus, Staphylococcus, and Disulfolococcus. Pyrrolysine and serine type proteases has temperature optima of 110 degrees centigrade and a half-life of 4 hours at 100 degrees centigrade. The serine protease from Sulfolococcus mucosus exhibit its activity at 100 degrees centigrade. Amylases. Amylases have been extracted from Pyrococcus furicus and Pyrococcus gaceae and are widely used in textile, confectionery, and brewing, paper, and alcohol industries. Glucose isomerase. Glucose isomerase is widely used in food industry. This enzyme converts glucose to fructose for use as sweetness. Cholineases. Cholineases too, obtained from Pyrococcus gaceae and Pyrococcus furicus, Thermococcus lithoralis, and Thermococcus celerus, Peridobacterium penniverum, and Sulfolococcus mucosus has temperature optimum of 105 degrees centigrade and is useful for bioconversion of starch into various useful products of industrial significance. Taq and PFU DNA polymerases. Taq and PFU DNA polymerases are used in molecular biology for the amplification of DNA using polymeric chain reaction. Taq polymerase found in Thermus aquaticus is active at 80 degrees centigrade at pH 8. Glutamase synthetase. Glutamase synthetase is an active at 100 degrees centigrade and is used for synthesis of glutamine from glutamate and ammonia. Aminotransferases. Two thermoactive aromatic aminotransferases have been purified and characterized from Thermococcus lithoralis and are active at 100 degrees centigrade. The enzyme aspartate aminotransferase that transfer amino group from glutamate to oxaloacetate has been isolated from Sulfolobus sulfactaricus. Molecular chaperones. Molecular chaperones are types of proteins that express under stress conditions such as elevated temperature and are involved in protein folding. They do not become a part of assembled protein but only assist in folding process. Molecular chaperones seem to be both extremely widespread and their sequence is highly conserved. Indeed, one important function of the molecular chaperone is to prevent improper aggregation of proteins. Molecular chaperones, in addition to folding newly synthesized protein, can also refold proteins that have partially denatured in the cell before protease recognize them as improperly folded and destroy them. Molecular chaperones have been isolated from Sulfolobus cibet and Sulfolobus sulfotaracus. So that is all about the molecular adaptation which is present in thermophilic organisms and hyperthermophilic organisms.